presentation. So good morning, everyone, or afternoon, depending on where you are. So today we were going to talk about sort of managing data in the laboratory, and it builds on Marianne's lecture from yesterday. And we're going to have a number of exercises. But what what we're trying to do is be able to create data products, right? So that the data can be easily utilized, right? So that if you want to make the data public available, or you're really following sort of open data principles, so that data is able to be freely used, shared, and built upon by anyone, anywhere, for any purpose. And this is becoming more important as more journals and more funders require people to make their data available with publication. And there are cases now, very journals that you submit to, so PLOS journals and others, if you have an article that is going to be published, you need to make that data available. Otherwise, they won't publish that article. But there are some problems. So, this is actually a funny sort of Twitter thread about overly honest methods. Who remembers, what you named, various Excel files, a couple of years ago. And again, if you don't have sort of standard practices that you sort of internalize and remember in terms of how you manage your data, things can get easily lost, sort of in the swamp of documents that exist on your computer or computers. So Marianne yesterday showed you this video about data sharing and various snafus. So I have a little spreadsheet here, the question is what does age mean? So anyone have an idea here what age is? Someone can unmute and, is it years, is it months, right? So this is an issue, in terms of having data that is documented properly, right? So I can't tell, for example, if this is years or months, right? It could be, an old mouse, in age in months, or or it could be an old human, age in years. And that's why making sure that the data is documented is important, right? And again, the same thing with time. What does that mean? In this case, it could be the start time. So this is like 11:59 a.m. It could be hours and minutes of something, right? Again, without actually knowing some of the specifics, it gets difficult. And the same thing with dir. What does that actually mean? And plat, with ones and zeros, right? So these are sort of undefined. But these, oops, let me go. Again, when you're sharing data, it's not just to make the data public. And so the description is important for many different reasons. So, if you're doing an experiment as part of your graduate work, you're going to be constantly sharing data with your PI, right? You may have multiple people collecting data for a study. So you have, maybe some undergraduate students that are actually running, various mice through the Morris water maze, and they're collecting data from you, and so you're seeing the data that they're providing. So you want to make sure that it's, completely documented. You may have different people doing data collection and analysis. We talked about blinding. And so here, you're going to have one group of people doing the collection, another group doing the analysis. So again, the people doing the analysis need to have a well-documented data set, and a well-structured data set, to be able to efficiently analyze the data. If you're a new graduate student in the lab, a lot of times coming into a lab, you're asked to do an analysis or look at some older data, right, to get yourself familiar with the type of data that's collected in the lab. Or, you may be doing sort of a meta-analysis or mega-analysis where you're looking across studies to say, what do these studies overall tell us? And during that pooling, again, you need to be aware of any of the differences, in the protocols, information about how the data is collected, how the data was captured, so that you can actually make decisions about what data you can use and you can't use. And again, this is all tied into the FAIR principles that Marianne described to you yesterday. And what we're talking about is that, the interoperable and reusable parts, are key, to making the data understandable. So those are the types of things that we'll sort of go through today. One of the things that came up with FAIR is that there are lots of different attributes to describe your data, right? So you have your data that is identified, with persistent IDs. Then there's sort of standardized ways in terms of representing the data in open file formats, and so that there's ability to get the data, and then there's metadata about the data itself, and that metadata can be a simple sort of core set of metadata that is then expanded upon by various domain-specific metadata. So, for example, we go back to the Morris water maze example. You have information about the mice, right? So what information about the mice do you have? Simple things are, age, sex, what strain it is, but there might be other types of data. So for example, if you're doing sort of work with knockout mice, so what genes, are affected in your various subject pools. So there's a lot of different metadata that can come in depending upon your experiment. And one of the places where one can go to actually explore, these standards, for different types of experiments, what type of, metadata is required is fairsharing.org. And so they provide, various collections of metadata standards, that are usable for various types of experiments. And so it's a good site, to be able to go to and browse those types of standards. And dkNET actually interacts with fair sharing. So when you look at our repository page, we actually link out to the fair sharing information about that, so you know what standards a database is using. So again, with fair data and the researcher, if you're looking at the researcher component, you're really focused on good data management, good documentation, rich metadata, preparing to share the data and submitting to a repository. And so we'll go through, these various aspects. This is really, something that starts at the very beginning of an experiment, right? So as you're beginning an experiment, you should already sort of have thought through, just as we're doing in these exercises today, some of the policies in terms of data management and documentation and metadata, right? So that should be done, before you actually start collecting any data, because once the data starts coming in, trying to reorganize it and get things straight, especially if you have multiple people collecting the data, can be very painful, because everyone sort of has a different idea of how they should structure data. So there is no structure defined beforehand. It's sort of the Wild West in terms of, what ends up on the hard disk. So starting with good data management, the basic idea for data management planning is to ensure that the data is high quality, well organized, documented, preserved, and accessible. And another key point is that if we have all this information, we should be able to, determine, some aspects of the validity of the data. And you might think of that sort of as quality assurance, quality control type processes on the data. However, a lot of times you're not explicitly taught good data management, right? There's there's no course, when I was going through my graduate career, no one taught us good data management. It was sort of just handed down from the person that you were working with. The metadata was sort of left up to the individual studies. um, a lot of researchers don't even know if they're collecting data and with the data sharing mandates, because they use boilerplate language. They're not familiar with the data repositories that they're actually going to have to place their data into once their studies are published. And because they don't know the repositories and the requirements, they're not familiar with the minimal information standards that they're going to have to follow. So again, that's why it's important to think about these data management practices very upfront. But there are motivations for why data management practices can be very helpful. As we all know, experiments are complicated and they can be organized in many different ways. So again, data management practices don't say that the data that you're producing has to fit one specific sort of set of guidelines, right? You can develop your own guidelines in terms of what works in your laboratory. That depends on how data is collected, who's using the data, and how the data is analyzed. We have to recognize that there are many files, many file types per subject. So again, going back to the Morris water maze, you may also, for example, if you're testing knockout mice, you may also have genomic data, so gene expression data, from those mice to determine, sort of levels of function that you can use in your analysis. Other issues are members of the same lab may use different ways to arrange data. So again, if you want to share data within the lab, there may be some difficulties there. And then, how does one share data, across sort of large scale projects, where a lot of data is being collected in various labs, around the world. And so the simplest first step is deciding on how to organize your files. Now, a lot of libraries are starting to get into developing information about some of these best practices. So for example, here I have, a link to the MIT Libraries data management section. So again, there are a lot of resources around about, to give you information about data management. So for example, here you can go to the MIT Libraries and find a whole section on that. Again, there are a number of different places that do this. And their basic set of strategies for your planning, highlight some important aspects, right? So spend time planning, the folder hierarchy and the file naming conventions. So again, don't think that you're going to be using all these data sets by hand, okay? One of the big things, that, you learn in graduate school, even in high school now, in some aspects, is developing scripts and pipelines, for example, via Python, to analyze your data in a consistent manner. To be able to do that, the files and where the files are located need to be in standard places for those scripts to be able to run through all that information. So for example, let's say you do your analysis, something looks off and you find out there's a bug in your code, right? You go back to your code for the script, you modify that script, now you have to run it across all your data set, all your data again. And again, you don't want to have to do that manually, so you want to be able to have that script be able to find, all the data it needs to analyze. So again, having this folder hierarchy and file naming convention is important. And again, there are lots of different variants on how one can do that. So for example, here, they give some examples, where they have projects, different experiments within the projects, and then, file types. You have, for example, the ability, to name your files with the date, the run type, and the sample type. So again, depending on your experiment, you need to come up with conventions that make sense, and that can easily be repeated by others that may be collecting data and analyzing the data. So I'll look at one example. So Open Euro was discussed yesterday. And so Open Euro uses the bids format, which I'll talk about in a little while. But basically, if you look at the dataset file tree on the right, you have a set of specific data description. So you have a data description document, you have a participants TSV, which lists all the participants in the study. And then you have, some information about some various tasks, and you have a folder for each subject, right? So you can start to see that, this data repository is using a standard structure. And the reason they do that is that when someone has the data in this standard structure, they can easily upload it to the database, and the database knows what to do with all that data, right? So the curation to actually get the data in the database is very minimal. And so again, the standards formats have various benefits, right? So we already talked about that the standard organization makes it easy to share data, within and across labs. If you have standards like bids, right, people actually develop software tools around these types of structures. So for example, if you're doing, some imaging work, and you don't have your data in bids format, there are a number of tools that, won't know what to do with that data, right? So you will have to convert that data into that format. Again, data repositories, depending again on the data that you're collecting. So for example, if you're collecting data from microarray experiments, you'll have to publish your data in Geo. So you should know up front, how you need to submit that data into Geo, what information, what metadata you need to collect, so that right up front, you're sort of creating what's needed to submit to a repository. Because if you wait till the end and then have to reformat everything, that's a lot of time that you have to sort of redo work to get data into that format. And again, if you have a good structured format, you can actually write a script to convert, one format to another. So again, even if your format does not exactly meet what a certain repository needs, if it's well defined and it's followed, you can easily create conversions, to do the depositions. And the other thing again is that analysis tools can be easily scripted. And as we discussed at the end of the exercise, focuses on raw data. A lot of repositories do require the raw data, but it's also important, to potentially include derived data and data that is used to do the analysis. So again, in the water maze example, there are different types of quote unquote raw data, right? So if you're doing the analysis of the study, the data that is used for that is actually the data that has been processed through a video processing pipeline that generates, the information about where the animal was during the trial, right? And so you generate the summary metrics off that you can do your statistics off that. But again, those metrics are derived from the videos. So you have these multiple tiers of data, that you need to be able to manage and, always provide links to, because the provenance of where you got that data in your spreadsheet, so for example, from the spreadsheet, to the analyzed file back to the video is important. So if you want some more information on bids, there's a number of things in terms of the bids specification that are online. But really what it's trying to do, it's trying to manage, the data and metadata through a neuroimaging workflow. And again, this is very similar to, the Morris water maze example, where you acquire data, you have some, raw data, you do some analysis workflows. You may come up with some information about, brain activation, for example, in a number of different subjects. And then you have to do some statistics across those activations that were derived from the data. And bids has its own sort of directory structure. Theirs is based sort of on a subject description. So you have a subject with a participant label. So that would be potentially the ID. So you'd have sub 01. You can have different sessions for that subject. So data for a session. So, with the Morris water maze, you may have, you do this over a number of days. So each of those days may be a session. So you can organize your data so that, that it's broken down so that you can find data from a specific day. And then you may have data from that session type divided into different types of folders. For example, as we said, you have the different data that's available. There's information you can store about the scripts you use in the code directory. You can also have results in this derivatives file, various stimuli. So again, in the water maze, there's no stimuli, but you can imagine that if you're doing some research where you're providing visual stimuli, that you might want to record that, and information about when certain visual stimuli were provided. When you look at sort of how this might look, there's sort of the subject information. There's, in this case, goes directly to the anatomy, because there are no sessions, for example, in this example. So it's just one session. So you may not need the sessions folder. And then different data files that sit below that. On the other hand, BIDS also provides a specification for a data set description. So how to describe the data set. Again, the database uses this to provide this information through the interface, but locally, it also provides a way for you to start collecting important information about this data set. For example, when it's being worked on. So you have the list of individuals who contributed to the experiment. You have a name, you have the funding information about how your research is funding, any information about publications related to the data set. So again, you can start building out a data set description around the data that you're collecting. In this case, instead of spreadsheets, there are other formats you can use. So again, when you're doing this in your own lab, it depends on sort of how your lab deals with data, what formats people are comfortable with. In this case, this is JSON, but it's very easy to read in the sense you have an attribute colon, and then the value for that attribute. So again, it's something that's, easy to read, different than a spreadsheet, but again, it all depends on, how work is done in your lab. And again, you can format this as a spreadsheet as well. So you do have, options there. For the participant files, again, these can be both, again, in spreadsheet, in this BIDS format can be spreadsheet or a JSON, and, they need to link up to the data, right? So you have the participant ID, this needs to map directly, to your directory structure. So it's important to make sure that your description files link directly to the directory. So you don't want to have a spreadsheet called control and patient where the subject ID is 1 in both of them, because it could be ambiguous. You have to read the name of the spreadsheet to be able to go back to, link it to the directory, which just complicates things unnecessarily. So make sure you use the exact participant ID that you're using in your directory structure. Then it makes your scripts or anything else you're generating very simple in terms of, how you relate one to the other. And again, in the spreadsheet, you're going to have multiple entries. You could have multiple entries for multiple dates and sessions in the spreadsheet. So it really depends, on what you're doing. And here again, we see sort of the issue. What is physio sampling, and what does that number actually mean? So again, if this is all that's uploaded to the system, someone may not be able to use this data because has no understanding of what that metric actually means. Within some of the work that we're doing, we're also looking to apply, the standard type of directory structure to other types of data. So, be it genomic information, be it microscopy, be it electrophysiology. And so we're building off sort of the BIDS format, and so again, you'll see very similar types of directory structures in terms of what we're doing. And when you go through the directory structure, in this case, we're going from the primary level. So you go from a subject folder into a session folder, right? And then you see that there are some derivatives based on some electrophysiology. So again, this is not imaging data. This is electrophysiology data, and we're using the same type of structures because they're fairly logical. And again, BIDS has been worked out over a number of years, and so you can actually use this, for this type of directory structure for other types of data. Again, not saying that this is going to be perfect, for whatever lab you're in, but it is something that is actually, usable, across a number of different domains. Because we're dealing with different types of files, so it's not just imaging data, some of the additional things we add, for example, to some of the directories, we add some manifest information, right? So that, this manifest can either be at the level of the data or it can be at a higher level, basically to describe the types of files that are contained in this data set. So again, because we're dealing with many different types of data, we don't have just one data type, so there may be, again, genomic data, there may be some microscopy data, there may be some electrophysiology data, there is data that is then, for example, various scaffolds or information about the structures that are also created based on some of this data. All this data exists, for example, in this same study, and people may not be familiar with it. So I think a couple days ago or yesterday, there was a question about the VCF file, right? What is a VCF file? So if you Google it, it's actually a calendar format. However, in genomic science, a VCF file is for documenting variants or variances in the genome. So it's a variant calling format, and so it lists these bits of information about the genomic data you have, and so it is a standard genomic data file. But if you're not from that field, you won't know what it is. So these manifests provide a way to describe that. And again, this is all, in the context of an NIH program. So this is a large program that's meant to collect data looking at the peripheral nervous system and how the peripheral nervous system relates to the potential for relieving conditions. So, how does modulation of the peripheral nervous system lead to potential useful treatments? And so, again, BIDS provides a base convention, right? We've modified that, for example. And again, there are other, in the genomic community, there are some, specific file conventions. So again, if you're organizing your data based on subjects, however you may be doing that, when you go down the folder structure, you may end up then in a genomic data folder that has its own structure, right? And that is defined by the various communities that collect that data. So again, this could be a mixing, of different types of standards that you use. And again, when you're developing things in your laboratory, important piece is this last comment. So how do you potentially extend, the file format when you are collecting different types of data, right? So if you're doing your experiment in your lab, collecting, one type of data, someone else in your lab is collecting data, looking at, the same sort of scientific questions, but has slightly different data that they're collecting, is there ability to sort of, create sort of a best practice so that, it's easy for people to go across these different types of studies. And then you need to define, what descriptor files you need. So in this example, you have a subjects file that describes a subject, but there's also a file about samples. So for example, if you're doing electrophysiology, you're pulling cells out of tissue, or if you're doing looking at genomic information from samples of tissue. Again, you need to document additional information for those samples. So again, this will depend completely on the type of experiments you're doing and what type of descriptive files you have. But make sure that you go through and describe each of these. And so we didn't talk about sort of the genomic data as much here, but some of those genomic data files or genomic data entities can contain thousands of files, right? And so, one of the things you also have to manage, when we're talking about these data formats is what the data formats entail. So if you have 1,000 files, you probably don't want to have thousands of files and thousands of files in the same directory. So you break that down into subdirectories, for example, when you're dealing with that. Okay, so we'll move on to the next set here. Here I'm going to talk a bit about good documentation and rich metadata. Data should be self-describing. So again, within the subjects files, you're going to have certain required things. So the ID, age, sex, group, species, strain, and then, for example, the RRID. And then there are, additional columns. So for example, husbandry, where did you get the animal? What is the body mass of the animal? So for example, if you're doing heart studies in pigs, you need the body mass, you need the units, and you need to know where you got the pig from. And so, those are additional types of information you might need to add. For samples, you need the sample ID, how that sample relates to a subject, where did you get that from? And then you're going to have additional columns to describe exactly how that sample was obtained and what that sample is about. When you collect your data, you're going to be collecting specific types of data, and you're going to need to then describe the data you're collecting. So if you remember back earlier in the presentation, we had spreadsheets with columns that were named things that are slightly ambiguous. The data dictionary is really meant to take those ambiguous column names and make them something that is very well documented. And so basically it's providing the metadata about the data elements. And when you start dealing with data dictionaries, you're going to start hearing about common data elements. And so data elements can be common across multiple data sets and different studies. And so there are different types of common data elements that you might encounter. They're universal common data elements. And so when you're dealing with specific fields of study, there may be standardized data elements for specific things that are required in your data set, including some demographic information about the study subjects. There are also then going to be domain-specific data elements. And so, if you're looking at, diabetes in a clinical setting, there are going to be certain data elements that you're going to be collecting that are going to be standardized so that the reporting is uniform. There are also going to be CDEs that are required by either a data repository or by institutional policy. And so you're going to have a mix of things that are required by the study itself, and then other information that you're going to have. And sometimes you'll hear these, with data repositories, that there are a set of core CDEs. So that there's a set of CDEs that are expected to be collected, right? So even if it's not part of your study, but your study is about a specific disease or something, and you actually have the ability to collect it, you may be required to provide that information so that across studies, right, that then there is sufficient metadata to do analysis across data sets. Okay, and so that's another thing when you're thinking about data you're collecting. It's data you're collecting for your study, but also what do you need to collect to ensure that your study fits in with other data within the repository. So that's why getting information about what standards you should be following and what repositories you're going to be using is important, because you need to make sure you're collecting the proper type of data. So NIH actually has been working on these CDE collections, and so there are a number of sort of broadly applicable and formally evaluated collections. So, for example, there's a FENX toolkit, which has a number of standard measures, for phenotypes and exposures. There's PROMISE, which deals with validated patient-reported outcome measures and has, a number of computerized adaptive tests, and the NIH Toolbox, which are validated measures of cognitive, emotional, sensory, and motor function. So again, these are data element collection processes which are being developed, and these also then tie into well-described data elements. There are also a number of other common data element working groups, around various NIH institutes. So, for example, within the Cancer Institute, within the Eye Institute, there are ophthalmic phenotype CDEs. NIDA, drug abuse, has substance and use disorder CDEs. So, as you sort of work through, also where, your funding is and things like that down the road, there are also going to be specific potential common data element libraries that you need to be aware of. And the other thing to be aware of is that common data elements are not so common. So if you go to, the common data element resource portal and you look at some of the databases that exist, some of them have over 250,000 common data elements. And the one thing to sort of be aware of here is that if there are slight variations in a questionnaire that you're giving someone, for example, if you're working with human subjects and you give them a questionnaire and you change one of the questions, that changes the whole questionnaire. And in a lot of database systems, that gets entered as a whole new set of common data elements. So, it's important to sort of understand how these common data elements are developed and, how that impacts sort of your reuse of them, if you are using them. There's also, various domain-specific metadata standards. So, for example, here, this is the MINDS experimental metadata, and you can see that there are a number of different, metadata elements that they describe. And again, these are some high-level metadata elements that would be useful for almost any study, that you can then work on. Again, these are just very sort of high-level metadata fields, even information to a protocol. So you heard about protocols.io a bit. You'll learn more about that next week, and so you can actually start using the protocols functionality within protocols.io to manage your protocols and link them back into your metadata specifically. And that's something potentially that you could do. When we look go back to sort of the data descriptions, there are other types of linkages to outside information for contributors. So your ORCID ID is sort of your identifier for research and is used by publishers, starting to be used by NIH. And so, want to make sure that you sort of bring in sort of links to these external bits of information, because it's sometimes hard to collect this information later on. So imagine you're doing a study in the lab and you have a graduate student, that is, working on the study with you. It's much better to collect that information about the ORCID ID and everything right then and there. Because what happens if they leave graduate school or, go on to a postdoc or go on into industry and haven't set up that ORCID, and then you have to somehow find them down the road once you're publishing your study, which could be many years later. So you want to make sure that you start collecting all this information again up front. And so this is just an example of sort of the data set description file. The other types of information that you might want to have, and this was brought up actually by one of the groups. So for example, if you have high level summary information, right? The other things you might want to keep for a data set, again, in terms of the summary information, is you might want to have sort of an example image of that data set. So if you're going to publish, for example, if you're going to publish that data set in some repositories, when they have sort of the informational page about that data set, they might require, sort of an image, right? So again, having some of this high level metadata can be useful because it's not just about the data in terms of the underlying data, but also when someone comes to a repository and is looking at your data sets, what type of information are you providing to them that is useful for them to determine if your data set is useful. So, when we talk about the manifest files, we can have, for example, a description of a file type, as I just described before. And that can either be the name of the file or a folder, or it could even be, a path descriptor or a data type folder descriptor. So if, for example, you have an ephys folder that is in every, subjects folder, right? You can describe that as being, containing electrophysiology data, and then the file type could be a description saying this is using the neurodata without borders format, right? So NWB is a standard electrophysiology file format. And so you can describe that there. And so this gives, the data set user, some knowledge about what's there. When you break that down, so again, what we're doing is, we then, you then have potential for data type folders. Again, so if you want to store different types of data, and especially if data has a lot of different types of files or multiple file types, within a data set. So for example, within imaging data, sometimes you have header files, sometimes, and you have the image files. And depending if it's a 3D volume, you may have one file per layer of that 3D volume. So you have a lot of files, and different files that relate to that. So you might want to be able to bundle those up. Again, this is just sort of some things that, with the work we do in curation, that becomes important. And again, if you're dealing with, sessions, you might have, subjects to sessions to data, or you might have just subjects to data if you have no sessions. You might have subjects, just a session if you have just one data type you're collecting. So again, it depends, again, on what your structure is. But if you then add sort of samples, right, you could have then, samples that also have sessions, or you could have the samples folder within the subjects folder. Again, lots of different variants here, but it needs to make sense for what you're doing. And so here you could see information if you have both subjects and samples, where you have the subject and the data type folders, and then the files for each of those subjects. And one thing that's important, one of the things that we found important, and this is also in the bid standard, oops, where's my little pointer here, is that the subject labels in the file names and additional, so having these sort of more complex file names can become important. Because yes, you have a directory hierarchy, but a lot of times in the scripts, you want to make sure that you're using the right files as well. And so, having that information in the file allows you to do that specifically within the script to pull, specific files, within an analysis. And again, you can have these manifests again at different levels, of the folder hierarchy. So you could put it in the data types, sort of way we described it before. But if you put it up higher, sort of in the sample level or the subject level, that is usually when you would be defining sort of standardized file naming conventions or folder naming conventions in terms of, what type of data should be expected. And the idea is that anything described at this higher level applies to, all the information at the lower level. The next section is about data collection. So here, you know, we want to deal with certain things that, you know, as you're pulling data in, you know, how do you identify missing data? So what happens if an instrument has issues, or, you know, someone didn't note the appropriate information? You know, you need to have some way to, you know, do a quality check to make sure that you're identifying this missing data. You know, how do you describe the missing data, right? So leaving a field blank doesn't really tell me what happened, right? You know, was it because it couldn't be collected, or whatever you're trying to measure didn't happen? So you need to specify specific data flags, you know, for describing this missing data. Again, define and enforce standards. So basically, you know, these standardized structures, naming schemes, layouts allow you to then develop tools to look at data quality, look for missing and anomalous values, and even if it's um, a study where you don't know what anomalous might be, if you use statistical techniques to allow to sort of, you know, generate information about any outliers. So if they're, you know, something that's, you know, very sort of out of the norm for the study at some point, especially when you're dealing with, you know, dozens and dozens of subjects, you know, you may be able to, you know, start detecting that something was wrong, or you know, using prior studies in terms of values that were generated, you know, to find these values. And you know, there are, you know, various data standards that are being developed. So, for example, you know, number of places, you know, USGS and others, you know, define, you know, rules by which data should be described and recorded. And this also deals with standardizing the format as well, in terms of formats that are used for data collection. And again, I'll go back to sort of my MIT libraries example, where they talk about file formats for long-term access. Right? So, for example, you know, for imaging files, you know, you want data that is lossless, right? So TIFF and JPEG 2000 are lossless formats. GIF and JPEG are not, right? So if you're storing your data for a long-term preservation, you want to make sure that it's, if it's uncompressed, or if it's compressed, that it's lossless. Also, that it's not encrypted, you know, because, you know, encryption keys can be lost and things like that. So, you know, we're looking for open, non-proprietary formats so anyone can get at the data. And for example, when you're dealing with tabular data, you probably want to store it in CSV, TSV, some ASCII format instead of Excel. I mean, Excel will probably last a while, but you can't guarantee, you know, especially with those file formats, that everyone has open access. You know, if they don't have Excel, and an Excel of a specific version, you know, to open up a spreadsheet, you know, if it was Excel 1992, you know, whereas if it was just a CSV file, you know, everyone has access to free tools to be able to get at that data. So the fairsharing.org also has information on these data formats. So again, a good resource for you to go to explore the different formats that repositories use. So, you know, and also the minimum reporting guidelines, right? So for animals in research, there's the ARRIVE guidelines. So, you know, animal research reporting in vivo experiments. So again, this is a guideline for, you know, the types of minimal information that you should collect. And so again, there's some of these community guidelines that are important. And then, for example, for specific experimental techniques. So if you're doing a microarray experiment, you're going to have specific minimal information you're going to want to store. The last bit of this is the controlled vocabularies that you will be interacting with and utilizing. And so here we're dealing about the terminologies and It could include ontology, so it could be just a standard controlled vocabulary, which is an alphabetical list of terms used, sort of with a hierarchical structure. So if you're talking about organs, kidney is an organ, so you have some relationship, just in terms of a hierarchy. Where ontologies include more complex interactions. So they can describe, for example, that certain cells, are located in the kidney, for example. And so they can connect terms to one another with these various types of structured relationships. Fair vocabularies, basically, we want to make sure that the vocabularies that are used also have some of the items of FAIR. So they have unique globally identified identifiers. And so you'll see in vocabularies and terminologies, ontologies, you'll see these identifiers that are either, they have sort of a short name up front and then an underscore or a colon and a number or some other sort of identifier. And that's a standard way to identify terms uniquely. And, again, they can have various different forms of how you spell that. In this case, anatomical area, or how it's described in terms of synonyms, acronyms. And so we make sure we need to make sure that all this information is stored, so that the different ways that people will refer to this, we can then associate that with this common term, so that no matter how someone annotates this in text, that it's easy then to link it to the ID, or if they provide the ID, it's easy to search based on how someone is familiar with how that term is labeled. And again, one of the things that's important is we want to make sure that there's human information, but also machine information. So FAIR talks a lot about machine accessibility. And so there are specific formats for terminologies that machines can access. So if you're writing scripts around terminology and metadata, you'll interact with OWL, RDF, some of these file types. And so what makes FAIR computable vocabulary? Well, it needs to be well documented. So definitions, how that term, has evolved, its provenance, the unique identifiers I mentioned, the identifier should be able to resolve to a landing page explaining that term. I mentioned the use of formal language, for the knowledge representation that can be accessed by computers, and then have other information, other important information, synonyms, abbreviation, acronyms, relationships, associated with that term. There's some global resources. There is the BioPortal, which browses across a number of different ontologies that you can use. This is up at Stanford. And then, we also within dkNET, we have an Interlex tool, and this basically, allows you to search for specific terms and then see, for example, what ontologies, so this term, for example, comes from the disease ontology. It also has information about relationships. So there are a number of tools out there for you to explore, terminologies, terms that you might want to include, for example, in your metadata. The other aspect of sort of working with data, especially in Spreadsheet, is that there are some best practices. So, one of the areas where you can get this information, there are a number of sort of data carpentry guidelines developed by different communities. This is one for ecologists, for example, and it has some tools about formatting data in spreadsheets. And so I'll give you a little example of, so this is actually a URL. So if I go to this URL, what you'll notice, is that, it goes through a whole number of different common spreadsheet errors, right? So, first one, do not use multiple tables in the same sheet. And we've encountered this in curating data, and it is very painful, because depending on how this is structured, the number of IDs, these things are going to change, and it makes it very difficult to actually access this data computationally. So if you're writing scripts or need to deal with it, right? Not filling in zeros. So again, this has a number of sort of best practices that you can go through. The other thing it talks about that I wanted to point out is quality control, right? Some simple so quality control doesn't have to be difficult. You can actually do validation within Excel and your spreadsheets, and this actually gives you a tutorial, on how to do that. And then at the end, it gives you exercises that you can do. So again, there are a number of resources out there that provide, assistance, in terms of working with spreadsheets. And data carpentry, there are a number of these courses that are given and workshops, and there's a data carpentry main site that links to a number of these different community developed tools. There are also other tools. So the Frictionless Data project, is really trying to develop methods to package, bundle, work with CSV files, and how you bundle the CSV files with appropriate metadata so that it's very easy for tools like R and MATLAB, SAS to bring in that data, without you having to do anything with it, right? So you have the metadata definition, you have your spreadsheet, and then these frictionless data tools will automatically use that to bring data into what tool you're using. And again, because we're running a little short, this is also a thought exercise. But again, you sort of, you started doing this in your document, and we can potentially get back to some of these in a number of weeks when we talk more about the data management plans more specifically. Oops, where is that? Okay. So the last thing that we were talking about was prepare to share. So one of the things that you have to start thinking about when you're sort of mapping this all out and when you're preparing to collect your data for future sharing is to identify the transformative steps in your data workflow. So, how is the data being collected? Where is it being collected? So for example, the raw data might be collected somewhere else. The data, that gets recoded, may then exist may be done on, a special computer. So for example, if you have a license for software, a lot of times that license is licensed per seat. So, there may be one computer in a lab that can do the recoding, right? So it doesn't exist on your computer. So again, you need to map out these workflows. What happens then with the screening and cleaning of the data, right? Because, you want to make sure all this data is in your study. So when we generated, the file folder descriptions before, you have all this data together, but in practice, as you're collecting that data, it may be distributed across separate systems. And so you will need to put into place steps to be able to bring that data together, so that you have a complete data set. And again, that's important to do before you start actually collecting the data. Run some tests, to make sure that what you're planning on doing actually works. If you're doing, various steps in terms of pre-processing, decide when you need to archive transformations. Is it just raw and finally processed, or is there a step in between? So let's say you're aligning data. Let's say you're doing imaging, of a specific organ, and you're trying to calculate size, or you're trying to extract calcium measurements. How do you align that information? Is there any information in terms of alignments where you have manual processes, in any of the pre-processing? So if anything's done manually in pre-processing, you probably want to take a snapshot of the data after you've manually pre-processed or done some manual intervention, because having to do that again is very time consuming. So again, think through this process. The other thing is, where do you then archive this data? So many universities have licenses with cloud storage. And so look at what your university supplies. So Google, UCSD, for example, has Google, and we can store unlimited data in Google for research. And so work with, what you're provided to do this archival, and you may have options within the lab as well. And so, we've talked a lot about the data and the data structure, and we'll follow up in 3 or so weeks, talking more specifically about data management planning tools. And so, just to sort of give an overview, the data management plans are really used to provide, everything we talked about today, everything from folder structures, how data exists, how, what file formats you're using, how you're archiving the data, and what are the policies for then the future access and sharing of the data. And how is this data going to be archived? So when you describe projects, for the NIH or NSF, you have to go through and document all this type of information for them. And so that's something we will actually work through in the follow-up.